So this is, I like this problem a lot. Again, I remember we had a whole lecture on deciphering the genetic code and all the wonderful things Gobind-Khorana did to decipher that code, and the repeating copolymer experiment was the key to deciphering the code, and it took some other scientists coming up with the random copolymer assay and the homo copolymer assay, and there's another technique, I think of a triplet binding assay that we didn't talk about too much because we don't have time to talk about everything. But let's just take a look at this problem so we can make sure we know how Gobind-Khorana went about his assays to finally figuring out the genetic code, confirming everyone else's results, and filling in any gaps that existed in the code at the time. So this problem looks at 3 different repeating copolymers. So the first one is a repeating dinucleotide of U and C. The second one is a repeating trinucleotide of UUC, and the third one is a repeating tetranucleotide of UUAC. Okay, so Gobind here will add these together and make a long RNA molecule. Same thing for these down here. And there's only a certain number of codons in these repeating molecules. Not a certain number, but a select, I guess a select, not a select number, but there's only a few codons in each of these repeating copolymers. Let's see if we can find them. So we have UCU here, and CUC, and UCU, and CUC, which are the same. So there's only 2 here. So UCU, remember the directionality is important. These are all 5' and 3' this way. And why is that? Because these are 5', 3', like this. So UCU and CUC. So 2 codons up there. And now here we have UUC, UUC. Okay, those are repeating. So we have UUC for one of them, UCU for another one, CUU for a third one, and then we start repeating again with UUC. Okay, so we have 3 types here. Now for a third one down here, okay, we have UUA, I can see UAC in there. I think there are usually 4 in a repeating tetranucleotide here. ACU, maybe not always 4, but all the examples I've seen, there have been 4. CUU, and then UUA starting again, so 4 different types in this one. Okay, so now what? We're not quite finished yet, but I've kind of drawn everything a little too big. So I'm going to select everything here, see if I can shrink it up down here just a little bit. Not like that, not like that, no. Okay, let me use the mouse instead of this pen. Oh, no. Like Napoleon Dynamite. That's my Napoleon Dynamite impression. It's terrible. Okay. Now what are we doing here? We are... Okay, so we haven't quite finished the question yet. So the question is, and again, this is on slide 20 of lecture 13, and it's listed, the question is listed under the suggested problems So I list the suggested problems for this lecture, and most of them are found from chapter 13, but this one right here is not found in the chapter. This one's just kind of printed on the slide. And we did go over this in class also, but just in case you missed the class or need some clarification, I'm doing it here. So let me see. So now the question gives us the proteins that are made when these repeating copolymers are used or added to the protein producing cell lysate. You know, and each one of those test tubes has a different radioactive amino acid added. Well, what we can find is that, you know, when this repeating copolymer here is added, we can get leucine added to a protein, and we can get serine added to the protein that's produced. Okay, for this one down here, this one leads to incorporation of phenylalanine, leucine, and serine. This last one down here, well, this one, okay, this only leads to three others: leucine, threonine, and tyrosine. So, you know, there are 4 codons, so two of these codons must code for the same amino acid, threonine. And so when Gobin Khorana is doing this experiment, you know, he doesn't know what codon goes with what codon yet. I mean, he doesn't have a little table like we do. He can't just, oh, that didn't work. He can't do what? What am I trying to do here? Hold on a second. I lost it. I wanted to paste in the genetic code and say, he can't just look at the genetic code and find out what the codons are. And no, he's trying to figure out the genetic code. Which codon goes with which amino acid. So, okay, so this is what we have, and this is what he's working with. He knows the sequences of the repeating copolymers because he made those. He's able to tell what amino acids are incorporated into the proteins when those repeating copolymers are used in the experiment. And he's got this data. So he knows the repeating copolymers. He knows the respective codons that go with those copolymers, and he's able to determine the amino acids that are incorporated into the proteins when those copolymers are used. So he's got this data, and he's just got to go through and, you know, like a puzzle, figure out which codon goes with which amino acid. And, you know, what the question says is, you know, with this information, you should be able to conclusively match 4 codons with their amino acids. List the codons with their matches below. Okay, let's see how can we do this. And usually, you know, I make a mistake, but I think I remember the easiest thing to do is, I should probably, no, okay, let's see if I can don't make a mistake here. So look for where we see the same codons. Do we have, okay, we have a CUU here and a CUU here. Okay, so we have to see the same amino acid. CUU, you know, the genetic code is unambiguous. The genetic code is unambiguous. That means one codon always codes for the same thing. If we give you a codon of this is CUU, it's always going to code for the same amino acid, or it's going to always code for a stop codon. In this case, it's an amino acid. So we need to look at CUU and see if we can find the same amino acid being incorporated into a protein in both experiments. So there must be an amino acid in common between these two. And there's only one, right? Leucine. There's a leucine down here, and there's a leucine up here. Okay, so we know CUU has to go with leucine. So I'm going to put this right here. We have leucine, 5' CUU. Okay, we've determined that one. Great. Now that we know CUU goes with leucine, we have 2 other, what color am I going to use? How about purple again? Now we have 2 codons here, UUC and UCU. Now, each of these has to go with either phenylalanine or serine. Phenylalanine or serine. One of these codes for phenylalanine, the other one codes for serine. Which is which? Well, what we can do is we can see we have a UCU here, and we also have a UCU up here. Okay, so UCU has to code for one, either phenylalanine or serine. So we need to see the same amino acid being incorporated into a protein down here as we see up here. Only serine is in common between these two. So UCU, because the genetic code is unambiguous, UCU has to code for the same amino acid every time. So it must code for serine, right? It can't code for leucine, and I'll explain that in a second. So UCU codes for serine because serine is found here and here, and UCU is found here and here. And we already determined that this UCU cannot code for this leucine in the previous part because it has to go with CUU here. So UCU here goes with serine, UCU here goes with serine. Okay, great, we found one for serine. So 5' UCU goes with serine. Okay, so where does that leave us now? Well, we need a different color. Okay, so we've matched up these two. Okay, so that leaves us with CUC. CUC, there's only one other amino acid that can correspond to CUC in this experiment. It's leucine. So CUC has to go with leucine. So we have another one for leucine. And this makes sense because the genetic code is degenerate. We can have more than one codon go to the same amino acid. So in this case, CUU and CUC code for leucine. I think there are 4 or 5 others for leucine. Now, okay, we have those two matched up with their amino acids, and down here we still have phenylalanine. I should use a different color. Use white. Phenylalanine here has not been matched up with anything, and there's only one more codon, UUC. Okay, so UUC has to go with phenylalanine. Phenylalanine, 5' UUC, 3'. Okay, great. So we have matched up 4 codons with 3 amino acids. And we can't really do anything with these two yet. So threonine and tyrosine are encoded by at least, one. Each one of these is encoded by at least one of these amino acids. So we don't know that, and we're going to have to do some more repeating copolymer experiments to figure this out. We can use this data. We still use this data and the new data that we can get from using new repeating copolymers. But for now, we can only match up these 4 codons with these 3 amino acids. Okay, so I hope you understand that, and I hope you think it is as fun as I do. Way better than playing Sudoku, however you say it, or some of those other puzzle games that are out there for wasting time and stuff like that. I think this is a lot more fun. Okay, so I will see you for the exam on Tuesday, and okay.